And good afternoon. It is August 17th, year 2023. My name is Mark Kulak, but I'm not going to be doing the presentation today. Today's presentation is actually just going to be an Audible book that I've been wanting to listen to. If you're like me, you have a lot of things in your hands, a lot of things going on. And it's just useful and helpful and pleasant to hear a good audiobook. And the book that we are going to be listening to today is Gene Machine, The Race to Decipher the Secrets of the Ribosome. It was written by Venki Ramakrishnan. He is a Nobel Prize winner, which can mean a lot of different things these days. But he is one of the foremost voices in the world right now in researching the ribosome. And the ribosome is the part of the cell which reads in RNA and produces proteins. The ribosome can be seen as the processor, if you will, of the cell. What is in our genes, which is in the DNA, is just code, and code can't do anything without a processor. Of course, the ribosome then, in turn, makes proteins based upon the inside code, internal code, the code of the cell itself, the construction of the ribosome, and a variety of other factors. And the shape of that protein determines what the protein will do or be used for, how the protein is folded, what it's made out of, so on and so forth. So it's not just code and a processor, it's actually multiple stages of it. But understanding how the ribosome works is the secret, is really the biggest challenge right now, and it's going to take a long time to figure it out, years, decades, perhaps. And I think I know my audience demands a lot, and I think that they are best served by understanding that while having mapped the human genome is an amazing accomplishment, it may be, they may have done not quite as much as they say, but is an amazing accomplishment. There's still so much more to be done to understanding how the machine of life works. And it may be unknowable, but if there is any chance of deciphering it, it has to start here. The work of Venki Ramakrishnan and the deciphering of the ribosome, it is my belief, and the belief of some others, is a big part of what is driving behind the scenes many decisions today in medicine, possibly even in global politics, potentially even I would go so far, I should say I might even go so far as to say it could be part of reorganizing the entire world around a model that could potentially provide the data to make this possible. So this is kind of a big deal. This book is listed as 8 hours and 11 minutes long. It came to my attention because it was this title and these concepts were actually promoted by a Gene Simon. And a Gene Simon is actually the executive director of President Biden's cancer moonshot movement. Simon has had various other roles. He was actually the executive director of the United States National Institutes of Health at one time. So I have some things to do right now on my own, so I benefit from this. This book is from 2019. I realize that this is copying material, but I think the importance of sharing the content with the audience outweighs, is worth it, okay? So with that said, this book may not stay online YouTube that long. We will see. I will probably keep versions of it for you to enjoy on other channels. I do encourage you, nonetheless, to purchase the physical book for your collection if it turns out to be a book of value. There may be things in here which could have been said better, or topics which are lightly pursued, and there may be some areas which we are going to learn a lot more about. Either way, I think it will be valuable. I'm deciding to invest my time, and perhaps you may be interested in choosing to invest some of your time with me. It's doubtful we will get this all done in one part. It will probably be 2, 3, or 4 videos. So with that said, I'm turning off my microphone. I'm clicking play, and then I'm going to get to work and you'll probably be doing much the same. All right. Take care. God bless.
Of the structure, a problem called non-identity.
And as I've already started it.
Chapter 5. Going forward.
This is Audible.
Highbridge, a division of Recorded Books, presents Gene Machine, The Race to Decipher the Secrets of the Ribosome. By Venki Ramakrishnan. Read by Matthew Waterson. Foreword by Jennifer Doudna. This is a personal story of the author's experiences as a student, professor, and avid experimentalist, seeking to understand how cells carry out one of the most ancient and fundamental of activities, the synthesis of proteins. The passion for discovery, the frustration of experiments gone awry, the personal and professional struggles that accompanied the road to scientific success come alive in this engaging account. The author's perspectives are unique in several ways. As an immigrant to America and later to England, and as a physicist entering the world of biology, the story captures the feelings of an outsider yearning to be part of the scientific and social world he observes. This yearning may have played a role in the approach to research that he describes. There is at once a desire to belong and yet a willingness to be a maverick and to embark on a journey of discovery that seemed at the outset to be a long shot. Then there is the scientific discovery itself. Revelatory structures of the very machinery that reads the genetic code and translates its nucleic acid sequence into chains of amino acids that form all of the proteins necessary for life on Earth. The ribosome, comprised of one large and one small subunit, is uncovered in all its glory, with the author's own work revealing the molecular underpinnings of the decoding mechanism of the ribosomal small subunit and the means by which multiple antibiotic drugs block the action of the bacterial ribosome and thereby eliminate microbial infections. The process of determining structures of the ribosomal small subunit, from initial efforts with individual components to later tour de force attempts to purify and crystallize the intact small subunit, is a fascinating tale of ingenuity, luck, and ultimate success. The story is also one of professional dilemmas, the serendipity of discovery, and the deeply human nature of research, in which personalities play an essential role. Any major scientific breakthrough involves multiple contributors, as well as the interactions of scientists grappling with the challenges of discovery and setback that accompany the winding path to understanding. It is not always clear how ideas emerge, whether they are truly one's own or instead an outcrop of discussions with others. Sometimes competition comes between colleagues and must be managed even as the joy of discovery is at hand. The award of the 2009 Nobel Prize in Chemistry gave recognition to the early success with ribosome crystallization by Ada Yonath and the subsequent achievements of Venki, Ramakrishnan, and Tom Steitz in solving the structures of the ribosomal subunits. Yet, as described in the book, others who made key contributions were, by the three-recipient limitation of the prize, left out of this recognition, despite the importance of their insights. The book provides a layman's account of the author's experiences and views on these matters that must be taken in the spirit of a memoir rather than an objective historical essay. Students of science and of the scientific method will find this story to be a fresh take on the process of discovery and the sometimes tortuous trail that leads to new knowledge. In the end, it is a fascinating contribution to the scientific literature, and one that will be valued as both a description of fact and a dissection of the emotional side of a scientist's approach and eventual achievement. J. A. Doudna. Prologue. Looking back, it is surprising how little effect her visit had. It was a grey autumn day in 1980. A small advertisement on a Yale University notice board had announced a lecture with a vague title. I had no trouble finding a seat, even though I arrived just before the speaker, because only a few specialists had bothered to come. She strode in exuding confidence, even audacity. After a brief introduction by her host, she began to describe the efforts of her group in Berlin to obtain crystals of an enormous assembly of molecules involved in translating genes into proteins. At the time, obtaining crystals was a key step to deciphering their structure. After her talk, there were hardly any questions because we weren't sure what to make of her work. We thought it was astonishing that someone had coaxed such a large and floppy particle to form the regular three-dimensional stacks of molecules that make up a crystal. As we walked down the hall back to our labs, a colleague of mine teased another, saying, "How come you can't even crystallize a tiny piece of it, and she's done the whole thing?" However, her crystals were not good enough to obtain a structure, and at the time, nobody even knew how to figure out the structure for something so large. So in the end, we thought it was an interesting curiosity, but none of us felt that our world had changed, and we needed to drop what we were doing. Little did I know that the scientist, Ada Yonath, would figure in my professional life for the next 3 decades, that I would compete with her and others in an intense race to understand an object that is at the heart of all life, or that one December I would be sitting between her and the Crown Princess of Sweden at a Nobel Prize banquet in Stockholm. Chapter 1. An Unexpected Change of Plans in America. When I left India, I had my heart set on becoming a theoretical physicist. I was 19 years old and had just graduated from Baroda University. It was customary to stay on to get a master's degree in India before going abroad for a PhD, but I was eager to go to America as soon as I could. To me, it was not only the land of opportunity, but also of rational heroes like Richard Feynman, whose famous Lectures on Physics had been part of my undergraduate curriculum. Besides, my parents were already there, as my father was taking a short sabbatical at the University of Illinois in Urbana. Since this was a last-minute decision, I had not taken the GRE exam that is a requirement for American graduate schools, and most universities would not even consider my application. The physics department at the University of Illinois initially accepted me, but when the graduate college found out I was only 19, they said that, at best, I could join as an undergraduate with 2 years of college credit. No middle-class Indian, then, could afford the cost of tuition and living in America. In the meantime, my department chair in Baroda showed me a letter from Ohio University asking him to let prospective graduate students know about its program. I'd never heard of Ohio University before, but saw that the department had an IBM System 360 computer and a Van de Graaff accelerator, and that its faculty had been trained at some of the best universities. That seemed good enough for me. They waived the usual GRE requirement and accepted me with financial support. After the typically nerve-wracking interview for a student visa at the US Consulate in Bombay, I bought my ticket to the Promised Land. As soon as I had finished my final exams, I left the sweltering heat of India and set off for America. I had caught a fever, and the flight seemed interminable, stopping at Beirut, Geneva, Paris, and London before landing in New York. I boarded a plane for Chicago, and then took a short flight to Champaign-Urbana. As I stepped out onto the tarmac on the evening of May 17th, 1971, I felt a blast of the coldest wind I had ever experienced. My sudden immersion into American college life came as something of a shock. College life in India was fairly staid. Students were conservative in their attire and focused on their studies. Many, like me, still lived with their parents. Dating, and especially premarital sex, was quite unusual. I arrived as a geeky student with a crew cut, glasses with thick black plastic frames, and orange suede shoes 2 sizes too large, into an America that, in 1971, was a continuation of the 60s. American students seemed to belong to an entirely different species. The males in tattered jeans and hair even longer than the females, who, in their hot pants and halter tops, seemed almost naked compared to the Indian women I had just left behind. Campuses throughout America were protesting against the Vietnam War. One afternoon, out of a mixture of curiosity and sympathy, I attended a peace rally. I stuck out like a sore thumb, but then found 2 older guys at the back, who, like me, had short hair and were dressed in the same cheap polyester trousers and shirts. I walked over to them and tried to be friendly, but they were rather curt and suspicious. Only later did I find out that they were FBI agents keeping an eye on the anti-war troublemakers. I spent the summer taking courses at the University of Illinois, filling gaps in my education from Baroda. At the end of the summer, I drove with my parents and sister to the pretty and hilly university town of Athens in southern Ohio, which was to be my home for the next few years. The first problem was finding any home at all. Since I had to live on my teaching assistantship and was a vegetarian, I thought it would be best if I rented a small flat where I could cook my own meals. We scoured the newspaper for rental ads with little success. In one case, the landlady said the flat was available, but when we showed up to look at it only a few minutes later, she took one look at me and said it was just taken. That was my first experience of racism in America. Unable to find a flat that weekend, I signed up for a dorm room and spent the first year living primarily on cheese sandwiches at the cafeteria. Notwithstanding its culinary disadvantages, the dorm room had the great benefit of allowing me to instantly acquire a group of friends and avoid the feeling of isolation and ghettoization so common to foreigners. My dorm mates quickly helped me assimilate into American college life. The first Saturday, we went to a football game. The pomp, with cheerleaders, bands, and the loud PA system, seemed to dwarf the game itself. The dorm also had the advantage of being close to the physics department, and several fellow graduate students lived in rooms nearby, so we were able to form a friendly study group and get used to graduate school together. Physics graduate students generally have to do a year or two of coursework, followed by a comprehensive exam before they begin serious research. Although I finished my coursework and the written part of the comprehensive exam without too many problems, the oral part at the end gave me the first inkling that I did not have a burning desire to be a physicist. I was asked what recent interesting discoveries in physics I had read about. I couldn't name even one, and only after some prodding could I even name an area I found interesting. They passed me anyway, and I decided to work under the supervision of Tomoyasu Tanaka, a well-regarded condensed matter theorist. By then, I had already become intrigued by biological questions, and I included some biological problems in my thesis proposal. Since neither Tomoyasu nor I knew the slightest thing about biology, these proposals were pure fantasy, and I soon abandoned them. As I began my thesis work, I realized that I could not quite see how to identify key questions, let alone how to approach them. Even worse, I didn't find my work interesting. I retreated into my social life, playing on the university chess team, going hiking with my friend Sudhir Kaika, learning about Western classical music from another friend, Tony Grimaldi, and generally doing anything except making progress on my graduate work. Tomoyasu was an almost stereotypically polite Japanese who would occasionally come into my office to delicately inquire about my progress, and I would tell him in a roundabout way that there hadn't been any. This continued for a couple of years. I've often said that if I'd had students like me, I would have fired them. Things suddenly changed when I met Vera Rosenberry, a recently separated woman with a 4-year-old daughter. Some mutual friends decided that the two of us should meet, perhaps because we were both vegetarians, an oddity in 1970s Southern Ohio. I was completely oblivious that our first meeting was a setup because we were part of a large Thanksgiving gathering. Observing my cluelessness, my friends decided I needed additional help and invited me to a dinner party with just one other couple. Vera struck me as both intelligent and classically good-looking, but I assumed someone like her would be out of my league and couldn't possibly be interested in me, so I tried to introduce her to a friend by inviting him to dinner along with Vera and her daughter, Tanya. I spent some of that time playing with Tanya so that my friend and Vera could be free to chat. It was my friend who had to point out that she seemed interested in me, not him, and if anything, she probably became even more interested when she saw how well I got along with her daughter. Despite this comically inept start on my part, we began a stormy courtship that lasted less than a year and got married soon after her divorce became final. At the age of 23, I found myself married and the stepfather of a 5-year-old girl. Marriage, however, focused my mind on my career. Vera wanted to have another child, so I faced the prospect of supporting a family without any idea of what I would do next. There seemed no question that if I stayed on in physics, I would spend the rest of my life doing boring and incremental calculations that wouldn't result in any real advance in understanding. Biology, on the other hand, was undergoing the sort of dramatic transformation that physics had in the early 20th century. The revolution in molecular biology that began with the structure of DNA was still going strong, and we were beginning to get fundamental insights into the molecular basis of biological processes that had puzzled us for centuries. Almost every single issue of Scientific American would report some major breakthrough in biology, and it seemed as if it could be done by mere mortals like me. My problem was that I knew only the most basic biology and had no idea what biological research entailed. So, even before I'd finished my physics PhD, I made the difficult decision to attend graduate school all over again, this time in biology, taking heart from the fact that many illustrious scientists like Max Perutz, Francis Crick, and Max Delbruck had made a similar transition. I wrote to several top-tier universities, but many of them did not want to accept someone who already had a PhD into graduate school. 2 replies were particularly memorable. The first, from Franklin Hutchinson at Yale, was a friendly letter saying that although they couldn't accept me as a graduate student, he would send around my CV to his faculty in case someone was interested in hiring me as a postdoctoral research fellow. 2 of them wrote back, Don Engelman and, ironically in hindsight, Tom Steitz. I thanked them both and said that I didn't have enough background to be useful to them as a postdoc and would try to acquire some training first. At the opposite end of the spectrum from Hutchinson was James Bonner of Caltech. In my applications, I had written that because I was only 23, I was young enough to go to graduate school again. Bonner berated me for bragging about my age, adding that he too was only 23 when he received his PhD and was considered backward in his family. He also said that the areas I had mentioned, allostery, membrane proteins, and neurobiology, were hardly surprising because they were the most fashionable areas of biology. If I wanted to work in them, he wrote, I had to first show that I could actually be competent in those areas, and certainly Caltech would not accept me as a student. Perhaps he had never read Catch-22. Fortunately for me, Dan Lindsley of UC San Diego was willing to accept me into the biology department as a graduate student with a fellowship. Even more fortunately, Vera and Tanya were game to move to California and continue living on a meager graduate student stipend with the added burden of an infant, all without a car. Somehow I scraped together enough work to produce a passable thesis just in time. Our son Raman was born just a month after my PhD exam. A couple of weeks later, a friend and I drove from Ohio to California in a Ryder truck with all our belongings, and Vera and the children flew in with my mother-in-law a week later. Once we had settled in, I began my studies in earnest in the autumn of 1976. The thing that immediately struck me about biology was how many facts you have to know. The introductory lectures for new graduate students were full of jargon I didn't understand at all. To catch up, I took a full load of undergraduate courses in genetics, biochemistry, and cell biology, in addition to doing first-year graduate student rotations, which are short, 6-week projects that American grad students typically do before they settle into a lab for their PhD research. Because my physics research had been entirely theoretical, I was totally ignorant of laboratory work. This was brought home to me during a rotation in Milton Sayers' lab, which worked on sugar uptake by bacteria. The experiment entailed adding a certain amount of radioactive glucose to a culture of bacteria at time 0, and then measuring how much glucose had entered the bacteria at various time points. The amount of the glucose to be added was much smaller than anything I'd encountered before, only about 20 microliters, less than 1% of the volume of a teaspoon. How could you measure such a small volume? I asked. The technician who was training me very pleasantly showed me a device called a pipetteman, which is essentially a tube with a piston that can be set so that it can go up or down by a precise amount. She showed me how to set the volume on the dial, how to draw up the right amount, and how to give the knob a little extra push at the end to make sure all the sample had been ejected. That was all there was to it, she said. I took the device and plunged it into the radioactive glucose, and she exclaimed, "What the hell are you doing? You have to use tips." The devices were so commonplace that she'd forgotten to mention the narrow disposable plastic tips that needed to be attached to the end of the pipetman so that it never got contaminated by contact with the sample. Moving with a young child and an infant was not exactly conducive to learning a new field. However, I was extremely lucky that Vera, who was starting her own career as a children's book illustrator, could work from home. She did almost all of the childcare and housework, allowing me to focus on my studies. I ended the first year feeling optimistic that I had a broad enough background in biology and fairly varied lab experience. In my second year, I began work with Mauricio Montal, who was studying proteins that let ions pass through the thin membranes of lipids that envelop all cells. As it turned out, I wouldn't stay in his lab very long. Almost entirely by chance, I would move across the country yet again to begin work on one of the oldest and most central molecules of life. Chapter 2. Stumbling into the Ribosome. Mention DNA and almost everyone nods in understanding. We all know, or think we know, what DNA means. It determines the essence of who we are and what we pass on to our children. DNA has become a metaphor for the fundamental qualities of almost anything. It is not in their DNA, we say, even when referring to a corporation. But mention the word ribosome, and you will usually be met with a blank stare, even by most scientists. A few years ago, I was told by Quentin Cooper on the BBC radio program Material World that the previous week's guest had been outraged that the eye only merited half a program when an entire episode was planned for a mere molecule like the ribosome. Of course, not only are most of the components of the eye made by the ribosome, but virtually every molecule in every cell in every form of life is either made by the ribosome or made by enzymes that are themselves made by the ribosome. In fact, by the time you hear this, the ribosomes in each of the trillions of cells in your body will have churned out thousands of proteins. Millions of life forms exist without eyes, but every one of them needs ribosomes. The discovery of the ribosome and its role in making proteins is the culmination of one of the great triumphs of modern biology. When I arrived in California to learn biology, like most physicists, I had no idea what the ribosome was, and only the vaguest idea of what a gene was. I knew genes carried the traits we inherited from our ancestors and passed on to our offspring. But I learned that genes are much more. They are the units of information that allow a whole organism to develop from a single cell like a fertilized egg. Although nearly all cells contain a full set of genes, different sets of genes are turned on or off in different tissues. So a hair or skin cell is very different from a liver or brain cell. But what actually are genes? Broadly speaking, a gene is a stretch of DNA that contains information on how and when to make a protein. Proteins carry out thousands of functions in life. For example, they are what make muscles move. They let us sense light, touch, and heat, and help us fight off diseases. They carry oxygen from our lungs to our muscles. Even thinking and remembering is made possible by proteins. Many proteins called enzymes catalyze the chemical reactions that make the thousands of other molecules in the cell. So ultimately, proteins not only give a cell its structure and shape, but also enable it to function. Understanding how the information in a piece of DNA could be used to make a protein was the culmination of an exciting decade that began with the classic 1953 paper on the double helical structure of DNA by James Watson and Francis Crick. Often, the structure of a molecule does not immediately explain how it works. Not so with DNA, which immediately suggested both how it could carry information and how it could reproduce itself. It had long been a mystery how information in a cell is duplicated when it divides, or how offspring inherit this information when an organism reproduces. In each molecule, the two strands of DNA that intertwine to form a double helix run in opposite directions. Each strand has a backbone of alternating sugar and phosphate groups, and one of 4 types of bases, A, T, C, or G is attached to the sugar and faces the inside of the helix. While playing with cardboard cutouts of the bases, Watson arrived at a brilliant insight. He realized that an A on one strand could chemically bond or pair to a T on the other, but not to any of the other bases, while a G on one strand could similarly pair with a C on the other. In doing so, the shape of each base pair, whether it was AT or CG, was about the same, which meant that regardless of the order of the bases, the overall shape and dimensions of the double helix was about the same. This formation of base pairs meant that the order of the bases on one strand would precisely specify the order on the other strand. When cells divided, the two strands would separate, and each would have the information to serve as a template to make the other strand, resulting in two copies of the DNA molecule from one. In this way, genes were able to duplicate themselves. After centuries, we finally understood in molecular terms how hereditary traits could be transmitted from generation to generation. The structure immediately suggested how genes could be duplicated and passed on, but not how the information in our genes could actually be used to make proteins. The problem was that each strand of DNA was a long chain made up of building blocks containing the 4 types of bases. But proteins are completely different chains made up of amino acids, and their chemical linkage is completely different. Their enormous variety comes from the fact that there are 20 types of amino acids, which have a wide range of chemical properties. The length and order of amino acids in each protein chain is unique, and amazingly, it contains the information needed for the chain to fold up into its own unique shape, which allows it to carry out its special function. Crick realized that the order of bases in DNA coded for the order of amino acids in a protein, but the question was how. Lots of people worked on this problem for well over a decade. It turns out that a stretch of DNA containing a gene is copied into a related molecule called messenger RNA or mRNA, so called because the molecule carries the genetic message to where it is needed. RNA, which stands for ribonucleic acid, differs from DNA or deoxyribonucleic acid by having an extra oxygen in the sugar ring. It too has 4 bases, but the base thymine, T, in DNA is replaced by a very similar base, uracil, U, in RNA, which, like T, also base pairs with A. How do you go from 4 types of bases to 20 types of amino acids? It would be like following a long sentence of instructions written in code using a foreign alphabet. It turns out that the bases are read in groups of 3 at a time, and each group is called a codon. The way they are read, something predicted by Crick, is that another RNA molecule called transfer RNA or tRNA has an appropriate amino acid attached at one end and a group of 3 bases called an anticodon at the other end. The anticodon and codon form base pairs just like the ones between the 2 strands of DNA. The next codon is recognized by a different tRNA, which brings along its amino acid, and so on. The next big discovery was that this doesn't happen by itself. Cell biologists discovered particles in cells where the mRNA is read and proteins are made. The particles were tiny by normal standards. You could pack 4,000 of them in the width of a human hair. There were thousands of them in every cell, from bacteria to humans, but they were enormous in molecular terms. Each of them contained about 50 proteins and 3 large pieces of their own RNA, a 3rd type of RNA, after mRNA and tRNA. Initially, scientists referred to the particles as ribonucleoprotein particles of the microsomal fraction because they were made up of both RNA and protein and were isolated from cellular fragments known as microsomes. This was quite a mouthful to say. So, in the late 1950s at a conference, Howard Dintzis suggested the name ribosome, which it has been called ever since. Dintzis was also the first person to figure out the direction in which a protein chain is made. Embarrassingly, even after working for 30 years in the field, I didn't know Dintzis or his work. When I finally met him in 2009 at Johns Hopkins University, where I had been invited to give a lecture named after him, he was still justifiably proud of having coined the word. The whole ribosome has over 1 million atoms. Because it is the link between our genes and the proteins they specify, the ribosome lies at the very crossroads of life. Even though everyone knew this, nobody had any idea what ribosomes looked like other than that they were blobs consisting of 2 parts. And that was a real problem. Somehow the ribosome bound mRNA and stitched together the amino acids brought together by the tRNAs into a protein. But without knowing what it looked like, how could we possibly understand how it all worked? Imagine you are a Martian, peering at Earth from above. You observe tiny objects on the surface that move mainly in straight lines, occasionally turning at right angles. If you were able to get a little closer, you might see that these objects move only when even smaller objects enter them and stop moving when they leave. If you have sensors, you could tell that they consume hydrocarbons and oxygen and emit carbon dioxide and water, along with some pollutants and heat. But you would have absolutely no idea what these objects really are, let alone how they work. Only by knowing the detailed construction of the object would you be able to see that it is made of hundreds of components that work together, and that it has an engine connected to a crankshaft that makes the wheels turn. You would need to see even more detail to know that the engine itself has chambers with pistons and draws in a mixture of fuel and oxygen that is ignited with a spark plug which drives the piston forward. It is the same with understanding molecules. Knowing the detailed structure of DNA revolutionized our understanding of how it works to store, transmit, and replicate genetic information. But the ribosome was not a simple molecule like DNA. It was enormous and complex and seemed just too daunting and intractable. Many great scientists like Crick, who had played key roles in figuring out how information in DNA is encoded, simply gave up on the ribosome and left for other fields. Sidney Brenner, an equally eminent colleague of Crick's, who was one of the discoverers of mRNA, said in the 1960s that the structure of the ribosome was a trivial problem and there was no need to work on it in Cambridge, since that sort of work would, of course, be done by Americans. This reminds me of Senator George Aiken saying of the intractable Vietnam War that the US should declare victory and get out. One of the early molecular biologists who persisted on the ribosome was Watson, who worked on the problem with Alfred Tessier, a biochemist from Geneva who was visiting his lab. Nearly 40 years later, at a meeting in Cold Spring Harbor in 2001, Watson recalled those early days, saying that when he realized how complex the ribosome was, he automatically knew that we would never know its structure. The ribosome was far from my mind as I settled into Mauricio Montal's lab. But after I had only been there a few months, I came across an article on the ribosome in Scientific American that would change my life. It described how to locate the many different proteins on the ribosome using neutron scattering, a technique known to physicists but hardly used in biology. The 2 authors were Don Engelman and Peter Moore, and I remembered that Don was one of the people who had expressed interest in having me as a postdoc when I was trying to switch from physics to biology. I thought if he'd wanted me with no biological background at all, he might be even more interested now that I had learned some biology and had over a year of lab experience. It also occurred to me that I had already learned enough biology to do research in the field, and that there was no need to get a second PhD in biology. So I wrote to Don, reminding him of our previous correspondence, saying I was now more prepared for a postdoc. Since I knew Don's main interest, like Mauricio's, was in membranes and proteins in membranes, I told him I would like to work on them in his lab. He wrote back, saying that he didn't have any positions, but his collaborator Peter Moore did, and if I came there and worked on ribosomes, I could then do some membrane work in my spare time. By that time, I knew that ribosomes were fundamentally important, so I said that was fine with me. As it turned out, the spare time was non-existent. Peter soon wrote, saying he was visiting San Diego for a conference and would be happy to meet me. When I went downtown to see him, he was dressed in his characteristically preppy clothes, with a brown corduroy jacket, and his thick glasses and manner completed the stereotype of an Ivy League intellectual, which indeed he was. He entered the fast track early in life and never left it, and I was never sure whether he really understood what it was like not to have been in elite institutions all your life. His father had made pioneering contributions to transplantation surgery at Harvard, and Peter himself had studied at a private school and then Yale before going to Harvard for grad school, where he worked with Watson on the ribosome. After that, he went to work with Watson's friend and collaborator Alfred Tessier in Geneva, who by then was a leader in the ribosome field. There, he set about purifying the different proteins that made up the ribosome. Realizing that the key to understanding the ribosome was knowing its structure, and that he needed to acquire skills in structural analysis, he left Geneva to go to the Medical Research Council MRC Laboratory of Molecular Biology in Cambridge, England. This lab was the direct descendant of the MRC unit where Watson and Crick had done their work on DNA, and by then had become a mecca for studying the structures of all kinds of biological molecules. Americans referred to the lab as the MRC, as if it was the only one of the many labs that the Medical Research Council supported throughout the country that was worth knowing about. The British called it the MRC LMB, or simply the LMB, as it is currently known. At the end of his stint at the LMB, Peter returned as a faculty member to his alma mater, Yale, where he has been ever since. He has a dry wit with a reservoir of knowledge that ranges from all aspects of science to history and the classics. Although a shy and reserved man, he lost his natural reticence when it came to science. His lectures were articulate and laced with humor, and generations of Yale students and scientists were exposed to his wrath when they presented sloppy arguments. At the conference in San Diego where we first met, he was standing by himself, waiting for me while people were milling about him. After a brief hello, we chatted about my background and his project. I wasn't at all sure how this informal interview had gone, but he wrote back soon afterwards to invite me to visit Yale. My visit there was very pleasant. Despite my obvious naivete, Peter formally offered me a position, and I accepted immediately. I spent the rest of the academic year finishing up work in Mauricio's lab. Finally, at the end of the summer, I left for New Haven, picking up my family en route in Ohio, where they had spent the previous few weeks. I arrived in Peter's lab in the autumn of 1978 with some trepidation. Faced with actually having to do postdoctoral research at Yale, my earlier confidence evaporated because, despite my 2 years of grad school in biology, I had very limited experience in actual biological research. A few days after I arrived, Peter and I were walking towards each other in a long passageway in the neo-Gothic Stirling Chemistry Laboratory. As soon as we drew close, he suddenly looked away. I was worried that he'd already regretted hiring me. But his longtime technician, Betty Rennie, laughed and told me that was just his manner. In any case, he was perfectly nice to me, and a year later, he must have felt I was sufficiently competent that he could leave me alone for an entire year while he went away on sabbatical in Oxford. During his absence, I grew a beard that I kept for almost 25 years. By the time I began working for Peter, some basic facts about the ribosome were already established. All ribosomes have two parts, known as the small and large subunits. The small subunit binds the mRNA containing the genetic information, while the large subunit actually stitches together the amino acids brought in by the tRNAs to make a protein. There are 3 slots for the tRNAs, one to bring in the new amino acid, one to hold the growing protein chain, and one that is kind of a halfway house before being ejected from the ribosome. During the process, the tRNAs move through the ribosome from one slot to the next, and as they move, they effectively drag the mRNA along with them, so that the ribosome is, in effect, moving along the mRNA, helping the tRNAs read one codon after another to make the protein. Each step needs the help of proteins that bind and leave the ribosome at various stages, and each step consumes energy. Because it uses energy and moves during this enormously complicated process, the ribosome is referred to as a molecular machine or a nanomachine. Apart from its fundamental role at the crossroads of biology between genes and the proteins they specify, there was a practical reason to be interested in the ribosome. Over the years, people had realized that many antibiotics work by blocking the ribosome at different steps. Because the ribosomes of humans are sufficiently different from those of bacteria, some of these antibiotics preferentially bind to bacterial ribosomes and are useful in treating infectious diseases. Bacteria are increasingly becoming resistant to infections, however, and knowing exactly how an antibiotic binds to the ribosome could lead to designing new and better ones. These basic facts had already made it into textbooks. So when I told people I was working on the ribosome, I would often be asked, "But isn't that already done?" Sometimes this would be accompanied by a pitying look, as though I was some poor guy dotting the I's and crossing the T's on a problem that was no longer interesting. The reality was that although we had an outline of what a ribosome did, we had no idea how it did even one of the many complicated steps involved in making a protein. It was as if we knew a bit more about a car, realized it had 4 wheels and windows and a driver who sat at a steering wheel, but knew nothing else about how it actually worked. Like many other fields, science has its fashions, and at any given time, some areas are considered more interesting than others. Often these are new areas where people are making rapid advances. Many scientists move on from a problem as soon as it gets too hard to make further progress. Those who are very creative open up entirely new areas, but others just follow one fashionable area after another. If everyone did this, our understanding of phenomena would be quite superficial. But fortunately, there are other scientists who stick with a problem, no matter how old and difficult it is, to get right to the bottom of things. Even though the ribosome had been studied for a couple of decades, nobody even knew where all the 50 or so proteins in it were located, let alone what they did. Peter was collaborating with Don Engelman to tackle this problem. In some ways, they could not have been more different. In contrast to Peter's reserve, Don was a tall, gregarious California native with a carefully manicured beard and a booming baritone voice and suave manner that conveyed great authority regardless of the topic of conversation. He had gone to Reed College in Portland, done his PhD at Yale, and then gone to do a postdoc with Maurice Wilkins, the third man of DNA, where he worked on the structure of the membranes that envelop all cells. Unlike Peter, who had spent his entire life working on one or other aspect of the ribosome, Don's interests were more varied. Don and Peter heard a talk by Benno Schonborn of Brookhaven National Lab about how neutrons could be used to study biological structures. Neutrons were something only physicists bothered with. Moreover, you needed a nuclear reactor to produce enough neutrons to do an experiment. But the interesting thing about neutrons for biology was that hydrogen and its heavier isotope, deuterium, interact very differently with neutrons, and hydrogen makes up half the atoms in biological molecules like proteins and RNA. The talk gave Don and Peter an idea for trying to figure out where the ribosomal proteins were. They realized that if you could somehow make a ribosome in which just 2 of the proteins had deuterium instead of hydrogen atoms, those 2 proteins would scatter neutrons differently. You could get deuterated proteins by growing bacteria in heavy water, which is deuterium oxide. Then you would have to reassemble a ribosome in which any 2 proteins of your choice were deuterated. Masayasu Nomura at Wisconsin had shown that you could biochemically extract the proteins from the small ribosomal subunit and separate each of the 20 proteins from the mixture by chromatography. You could then mix all the components together in a solution, and, under the right conditions, you could reassemble a functional small subunit from the purified proteins and RNA. This meant that you could make a small subunit in which just 2 of the proteins were replaced by their deuterated counterparts. These ribosomal subunits could then be taken to a nuclear reactor in Brookhaven National Lab in the middle of Long Island and exposed to a neutron beam. Each such experiment would give you a distance between a pair of proteins. By measuring distances between lots of different pairs, you could figure out how they were arranged in 3 dimensions, very much like how early surveyors mapped unknown terrain by triangulation. The project involved tediously making the same kinds of measurements over and over again with different pairs of proteins in the ribosome. I joined the lab when only the first few of the proteins had been located this way, and the baton was handed to me by the previous postdoc Dan Schindler. To my surprise, I learned that neutron beams, even from a nuclear reactor, are orders of magnitude weaker than X-ray beams, so it would take several days to measure the small signal from the deuterated proteins that was buried in a large overall background scattering from the rest of the ribosome. Doing this work during the summer had advantages. While the data were being collected, I would occasionally go to the beach on Fire Island, a few miles directly south. At other times, being trapped in Brookhaven was not exactly thrilling, because the lab was in an old army camp in the middle of nowhere outside Yafank. The scientists who worked there lived in communities miles away. They were a mixture of rural hamlets and extended suburban sprawl. Unlike a university town with a thriving cultural scene and nightlife, the lab was deserted on evenings and weekends, with nothing for a temporary visitor to do. This situation reminded me of a famous New Yorker cartoon of the Long Island Expressway that said, "Exit 66, Yafank. If you have already been to Yafank, please disregard this exit." After about 3 years, a little over half of the proteins were placed in the small subunit, and we wrote a couple of papers on their location. I was wondering how long it would take me to do the remaining ones, but as I approached the end of my fellowship, Don came to me and said it was in my own interest that I move on to the next phase of my career now that I had acquired the training I needed as a postdoc. The project was eventually finished by my successor, Malcolm Capel. The final paper describing the locations of all the proteins showed them as billiard balls superimposed on the shape of the small subunit, and I would joke that about a third of those balls were mine. Responding to Don's hint, I applied for almost 50 faculty positions, but it was not a great time to be looking for jobs. We had just entered the Reagan era of minimal government, and research funding was tight. Biotech was still in its infancy, and faculty jobs were scarce. I applied for every opening from junior colleges to universities of varying quality. The smaller teaching colleges looked at my long Indian name and probably worried I couldn't speak English well enough to teach. The universities looked at my career, a bachelor's and doctorate in physics, neither of which was from a prestigious university, 2 years studying biology without a degree, followed by research using a technique nobody had heard of to work on an old problem that was already unfashionable. No wonder I didn't get a single interview. Luckily, Oak Ridge National Lab in Tennessee had just started a neutron scattering facility and was looking for someone to collaborate with biologists. So Don called up the head of the facility, Wally Kohler, to recommend me. In the excitement and optimism of starting my first real job, Vera and I bought a house there right away. In February of 1982, we packed our stuff into our small Ford Fiesta and drove from New Haven to Tennessee through an ice storm in Pennsylvania. I had gone there on the understanding that I would be able to do my own research, but when I arrived, the biology lab I was promised didn't materialize. Wally Kohler told me that I was there to collaborate with biologists on neutron scattering, not to do my own research. He was a well-known physicist whom I liked personally, but I felt he did not understand the way biology is done and the rather peripheral role neutrons play in biology, and I started looking to leave Oak Ridge very soon after I got there. Luckily for me, Benno Schönborn, who was developing the biological use of neutrons at Brookhaven National Lab and who had inspired Peter and Don to work on the ribosome, came to the rescue. He offered me an independent job at Brookhaven, which, given my situation in Oak Ridge, I was grateful to accept. So, only 15 months after our arrival in Oak Ridge, we sold our house for a substantial loss in the summer of 1983 and moved back to the East Coast, this time to Long Island. Vera was not happy at leaving her beautiful garden and idyllic life in Oak Ridge, and her heart sank when we drove over the George Washington Bridge and saw all the traffic on the Long Island Expressway. We eventually found a house in East Patchogue, right next to the village of Bellport, on the south shore of Long Island. It was a 12-mile commute each way to the lab and seemed much longer during the ice storms every winter. Unlike my disastrous experience in Oak Ridge, Brookhaven gave me a well-equipped lab, a technician, and freedom to carry out my own research. My colleagues were very friendly and helpful, but made it clear that I shouldn't expect to get tenure for merely continuing work I had done as a postdoc. Fortunately, As a result of collaborations during my brief stint in Oak Ridge, I had developed an interest in chromatin, the complex of DNA and histone proteins that makes up chromosomes in cells. So I began work on how chromatin was organized, and for a long time was better known for this than for my continuing ribosome work. I still kept plugging away at the ribosome using the techniques I had learned, like neutron scattering, but neither I nor anyone else in the field made real progress in understanding how the ribosome actually worked. Individual components of the ribosome seemed to do little by themselves. It was a bit like looking at a set of tires and pistons in isolation, with no idea of how they were put together to make a car. On the other hand, the whole ribosome seemed too large and intractable a problem on which to make any headway. Not only was the ribosome becoming even less fashionable than when I'd started working on it, but neutron scattering was proving a dead end in tackling it or chromatin. Almost a decade after I'd switched from physics to biology, it looked like my second career was going down the tubes, just like my first one. Chapter 3. Seeing the Invisible. We say seeing is believing, and it is astonishing how often just being able to see things has changed our understanding of the world. For centuries, we had many misconceptions about our own bodies because our knowledge of them came from the Greek physician Galen, who based it on the dissection of animals. It was only in the 1500s, when Andreas Vesalius started dissecting human corpses, that we started to understand our own anatomy. When it came to seeing the ribosome, however, none of the methods we were using will let us visualize details of the ribosome, let alone how it worked. Before returning to our story, it is worth making a detour into how scientists took half a century to develop a technique that would be key to cracking the ribosome problem. For most of human history, we were limited by what we could see with the naked eye. The detail we could see dramatically expanded in the mid-1600s when a Dutch linen merchant, Antony van Leeuwenhoek, wanted to examine cloth fibers more closely. In his desire to make better lenses, he would go on to invent the most powerful microscope of his time, which he used to look at everything from pond water to the scum scraped off his own teeth. He was astonished to see small creatures moving around, which he called animalcules, but which we know today as microbes. Soon afterwards, Robert Hooke also used microscopes to look at the details of everything from fleas to various tissues. He coined the term cell to describe the compartments that made up plant tissues. The idea of the cell completely transformed biology. We now realize that the cell is the smallest unit of life that can exist independently. And cells can associate to form tissues and whole animals. As microscopes became more powerful, people saw that the insides of cells had structures, such as a nucleus with chromosomes and various organelles. Just being able to see detail had transformed biology from the anatomy of humans to the structures inside a cell. But what were all these things inside cells made of? Like all everyday matter, cells and their components are made of molecules, which are groups of atoms that are held together in a very specific way. The atomic theory of matter took such a long time to develop and is so important that Richard Feynman, the famous physicist, said that if all scientific knowledge were to be destroyed and only one sentence could be passed on to the next generations of humans, it should be, "All things are made of atoms, little particles that move around in perpetual motion, attracting each other when they are a little distance apart, but repelling upon being squeezed into each other." It is astonishing that in the 18th and 19th centuries, without ever being able to see molecules, scientists not only deduced their existence, but also their structure, the arrangement of the atoms that make up the molecule. They could do this for simple molecules like common salt, which only has 2 atoms, and somewhat more complicated molecules like sugar, which has a couple of dozen. But with larger and more complex molecules, it becomes increasingly difficult to infer their structures without being able to visualize them directly. The reason nobody had ever seen a molecule has to do with the properties of light itself. Light is made up of photons, which we know from quantum physics can have the properties of both particles and waves. The wave-like nature of light is the reason lenses and microscopes work. But this property also means that when light passes through a very narrow opening or around the edge of an object, it spreads out because of its wave-like nature in a process called diffraction. Normally, we don't notice this effect. But if two very small objects are close together, the images spread out and merge with each other, and someone looking through a microscope would just see one large fuzzy object rather than two distinct ones. In the 19th century, the German physicist Ernst Abbe calculated that you could only see two objects as separate or resolve them if they were no closer than half the wavelength of the light used to look at them. Visible light typically has a wavelength of 500 nanometers. A nanometer is 1/1000th of a meter. Any features that were closer together than about 250 nanometers wouldn't be seen as separate. This minimum distance between two objects that could be seen as distinct or resolved is called the resolution limit. When using light, very fine details, such as features that are closer than 250 nanometers, simply wouldn't be seen, but would be blurred out. People had estimated by the early 20th century how many molecules there would be in a volume of material, so they knew the approximate distance between the atoms in a molecule. It turned out to be over 1,000 times smaller than the wavelength of light. This meant that it would be impossible to see individual molecules, even with the best light microscopes. Molecules would be forever invisible. An alternative to light emerged when a curious new radiation was discovered in 1895 by Wilhelm Röntgen, a German physicist who was looking at discharges from vacuum tubes. These tubes have two electrodes separated by a high voltage in a vacuum. When a current is applied, the negatively charged electrode, or cathode, heats up and emits electrons, which then fly through the vacuum and hit the other electrode, the anode. He discovered that these tubes emitted mysterious rays that caused a barium compound to glow even in total darkness. He called these mysterious rays X-rays and started to investigate their properties. They were highly penetrating and allowed people for the first time to see through normally opaque objects like the human hand, revealing the bones inside. Nobody really knew what X-rays were or even whether they were particles or waves. We now know that they too are photons like ordinary light, so they are both particles and waves. In 1912, Max von Laue and two of his associates decided to see what would happen when X-rays hit a crystal of zinc sulfide, which consists of two types of atoms, zinc and sulfur. They found that the X-rays, instead of being scattered all over, were concentrated in spots. Von Laue quickly realized what was happening. He had used a crystal, which he realized was just a regular three-dimensional arrangement of molecules, like a stack of perfectly spherical balls. When X-rays hit the crystal, if they were waves, each atom would scatter the waves in all directions, just as when you throw a pebble in a pond, waves ripple outward in every direction. The resulting wave in any direction would be the sum of all the waves scattered by each atom that had been hit by the X-ray beam. When two waves combine, the strength of the combined wave depends on how the two original waves add up, which in turn depends on how they are related to each other. If they each have their peaks and valleys in the same place, they are said to be in phase, and the combined wave will be twice as strong. But instead, if the peaks of one line up with the valleys of the other, they are out of phase and will completely cancel each other out. Anywhere in between will produce an intermediate result. Von Laue realized that, depending on where each atom was located, the waves scattering off them would travel different distances. They would lag behind or be ahead, so they would no longer be in phase and would largely cancel each other out. But in certain directions, the waves from the different atoms would lag or be ahead by a whole number of wavelengths. In that case, their peaks and valleys would still line up, so they would still be in phase and reinforce each other. That's why von Laue saw just spots in his photograph. They indicated the directions in which the wave scattering off the atoms in the crystals had reinforced one another. The experiment showed that X-rays could certainly be thought of as waves, but it was also the first direct proof that a crystal consisted of regular arrangements of atoms. Since people knew roughly how far apart atoms would have to be, this also told them what the wavelength of the X-rays was. it was just right to see atomic detail, more than 1,000 times smaller than the wavelength of light. 2 years later, in 1914, von Laue was awarded the Nobel Prize in Physics. Von Laue had also tried to deduce exactly how the zinc and sulfur atoms in his crystal were arranged in space. But here, his analysis turned out to be wrong. In Cambridge, a young graduate student named Lawrence Bragg was intrigued by von Laue's results. He puzzled over them and came up with an elegant way of looking at the problem that helped deduce the correct structure. Bragg realized that the atoms in a crystal could be thought of as forming different sets of planes. These sets of planes could be in different directions and separated by different spacings. The X-rays scattering off the atoms in such a plane could be thought of as reflecting off the plane, so the diffraction spots are also called reflections. For any set of planes, the additional path traveled by X-rays scattered off adjacent planes is a whole wavelength for a particular angle. For that angle, the waves scattering from each set of planes will stay in phase and reinforce one another, giving rise to a diffraction spot. The relationship between the angle and the distance between the planes is called Bragg's law. At any given position, there might be several planes that satisfy the Bragg condition, each giving rise to a spot at a particular angle relative to the incident X-ray beam. Also, as you rotate the crystal, new planes will satisfy the Bragg condition and give rise to more spots. When you've completely rotated the crystal about the beam, you will have measured all the possible spots from the crystal. Using his analysis, Bragg worked out the correct arrangement of the atoms in von Laue's crystal. He reported his analysis to the Cambridge Philosophical Society in November of 1912, but because he was only a student, his professor, J.J. Thomson, who had discovered the electron, had to officially communicate the article Bragg wrote for the Society's journal. Bragg then used his theory to analyze one of the simplest molecules around, common salt. By then, chemists had concluded that a molecule of salt consisted of a sodium atom and a chlorine atom held together, which they called sodium chloride. When Bragg analyzed the spots from his X-ray photographs of salt crystals, he found that there was no sodium chloride molecule. Instead, the crystal was a three-dimensional chessboard arrangement of sodium and chlorine ions, in which the sodium ion has lost an electron and the chlorine atom has gained one, so the two have opposite charges. The ions are then held in the crystal by electrical forces. Many chemists of his time didn't take kindly to a young physics graduate student telling them that even a simple thing like common salt wasn't what they had thought it was. One of them, Henry Armstrong, a chemistry professor at Imperial College London, viciously attacked Bragg in a letter to the journal Nature titled Poor Common Salt, saying that Bragg's structure of sodium chloride was more than repugnant to common sense. In what was perhaps the ultimate insult to an Englishman, he added, "It is absurd to the nth degree, not chemical cricket." In the end, Bragg not only turned out to be right, but went on to determine the structures of lots of simple molecules. For the first time, molecules could be seen. This method of determining the three-dimensional arrangement of atoms in a molecule by getting them to form crystals and analyzing the diffraction spots came to be called X-ray crystallography. Bragg's father, William Bragg, actually they were both William, so the son used his middle name Lawrence, was a professor of physics and had developed some of the most advanced instruments of his time to make accurate measurements of the X-ray spots. Once Bragg had worked out the theory, he and his father worked together on several experiments. While Bragg remained in Cambridge, his father, who was already a famous physicist, was traveling widely, giving lectures on his work with his boy. For some time, Bragg was worried that since he was a mere student, his well-known father would get all the credit for their work, and there was apparently some tension between them. As it turned out, someone on the Nobel Committee was very well informed. In 1915, both Braggs shared the Nobel Prize in Physics for their work. Bragg, who was then 25, remains the youngest science Nobel laureate. He could not go to Stockholm to receive the prize because World War I had just started. In fact, Bragg's brother Robert had been killed in action only a few weeks before he heard of the award. Bragg ended up delivering his Nobel lecture in 1922. The simple molecules that Bragg initially studied had only a few atoms, so it was possible to try out different guesses for their structures and see if the spots predicted by Bragg's law would match up with what was actually seen in the photographs. But this sort of guesswork became more and more difficult for larger molecules that had many more atoms. At this point, a different approach was needed. Could you directly calculate an image or map of the molecule from the X-ray data that would show you directly where the atoms are? To understand how calculating a map works, think of how a magnified image is obtained using a lens. Light rays are scattered from each part of the object. Each point on the image is produced by having the lens combine the scattered waves from every point on the object. The important thing is that the scattered rays exist whether a lens is there or not. The lens simply collects them to form an image. We've discussed how the wavelength of light is almost 1,000 times too large to see atoms in a molecule. X-rays, on the other hand, have the right wavelength. Couldn't we just use X-rays with a lens to see images of molecules directly and not bother with crystals and spots? The problem is that there isn't a good enough lens to make images of molecules with X-rays. But even if we could do that, there is a serious problem because, unlike light, X-rays damage the molecules they hit. To see an individual molecule in sufficient detail, you would have to expose it to such a high dose of X-rays that you would end up destroying the molecule. In a crystal, however, the diffraction spots are the result of adding up the scattered X-rays from millions of molecules. The amplified signal from these millions of molecules means that you can get away with a much smaller dose of X-rays and is another important reason to use crystals. Without a lens for X-rays, people figured out clever ways to do mathematically what a lens does: combine waves from different parts of the object into an image. For the mathematically inclined, this is doing a Fourier transform of the scattered rays. But there was a big problem with just taking the measured spots in an X-ray photograph and combining them in a computer to form an image. A lens knows when each part of a wave has arrived when recombining it with others. In other words, the lens knows the phase or relative position of the peaks and valleys of each of the waves it has to add up. When we measure the intensity of an X-ray diffraction spot from a crystal, what we are measuring is the amplitude of the wave. In other words, the height of its peak above its average position. The measurement has no information at all about the phase of the wave, which is how much farther ahead or behind the peak of the wave for each spot is relative to all the others. To add up the waves corresponding to all the spots, you need both pieces of information. But the measurements contain only half the data. To make matters worse, it is the less important half because the image is much more sensitive to having the right phase than having the right amplitude. This vexing situation is called the phase problem in crystallography. Without knowing the phases, there would be no image of the structure. The crystallographer Arthur Lindo Patterson came up with one way around the problem when he realized that, even without phases, you could use the measured intensities of the spots to calculate a function that would allow you to locate the most prominent of the atoms in the structure, which typically were the heavier atoms, because they have more electrons and scatter more. You could then calculate the phases that just these atoms would give rise to, and combine them with the actual measured amplitudes from the whole molecule. When you do this, some of the missing atoms, the ones that were not part of your initial few atoms, show up as weaker features or ghosts in the image of the structure. When you add those atoms to your initial structure and redo the calculation, then even more atoms show up as ghosts in the next round. In this way, you can gradually bootstrap your way to the final complete structure. The end result of this is a three-dimensional image or map of the molecule. These maps are called electron density maps because almost all the scattering of X-rays is from electrons in the atoms, so the maps show how dense the electrons are at any given location. In practice, since the electrons mostly form a tight core around the nucleus, this method tells you where the atoms are. The maps are visualized by making contour maps of sections, similar to topographical maps that show where mountain peaks are. In topographical maps, the higher the altitude, the higher the contour level. In electron density maps, the greater the density, the higher the contour level. So the maps show where the atoms in a molecule are. Scientists started using the Patterson method to determine the structure of increasingly complicated molecules. One person who pushed this method to its limit was Dorothy Hodgkin, née Crowfoot. She was one of the first women to get a first-class honours degree in chemistry from Somerville College at Oxford University, and then went to get her PhD working under John Desmond Bernal in Cambridge. Bernal was a brilliant polymath, but he was something of an intellectual butterfly. He often made the first pioneering forays into lots of important problems, but didn't always see them to completion. Perhaps he had too many distractions. During World War II, he was asked by the British government to advise them on the best sites for the D-Day landings in Normandy. He was an ardent communist and remained an apologist for the Soviet government even after Stalin's atrocities became known. He was also ardent about women, on occasion having relationships with several of them at once. Many of them, including Hodgkin herself, sensed that Bernal genuinely cared about them and encouraged them in their careers, and remained on very good terms with him for the rest of their lives, long after they were no longer involved with him. In fact, several of them took turns taking care of him when he was terminally ill. Perhaps as a result of his various distractions, several of Bernal's protégés went on to make much greater contributions and become more famous. Hodgkin was one of the most illustrious of these. She returned to Oxford after her PhD, but the academic world was not at all receptive to hiring women, and she was unable to get a proper faculty position there. Luckily, her old college, Somerville, gave her a fellowship, which she supplemented with various temporary research grants. She was given space in the attic of the university's Natural History Museum, and to carry out her experiments there, she frequently had to precariously balance her precious crystals in one hand while she climbed up a ladder. Undeterred by her difficult and uncertain working conditions, she showed exceptional judgment in choosing to work on important biological molecules, including penicillin and vitamin B12. The latter contained several hundred atoms, and the determination of its structure was considered a tour de force. At one point, Bernal told her she would win a Nobel Prize. She asked whether she might one day be elected a Fellow of the Royal Society, and he is reported to have said, "That would be much more difficult." For men, it would have been the other way around, but at the time, the Royal Society had not elected any women in its nearly 300 years of existence. But Hodgkin's work was simply far too important to ignore. She was made a Fellow of the Royal Society in 1947, just 2 years after the Society welcomed its first female Fellows, the crystallographer Kathleen Lonsdale and the biochemist Marjorie Stevenson. In 1964, Hodgkin went on to win the Nobel Prize for her work, an event reported with the headline, "Nobel Prize for a Wife from Oxford," beginning the article with, "A housewife and mother of 3 yesterday won the Nobel Prize for Chemistry." Clearly, for at least some journalists, her domestic status and ability to procreate were still the most important facts about her. X-ray crystallography had been a resounding success, but it wasn't clear initially that molecules like proteins could be studied by crystallography at all. In the mid-1930s, when Bernal and Hodgkin first examined crystals of a protein with X-rays, they hardly saw any spots. Bernal realized that protein crystals contained quite a lot of water and were drying out and losing the regular arrangement they originally had had. When he and Hodgkin kept them hydrated while doing their experiments, they saw a beautiful diffraction pattern. It was the first proof that proteins could have a defined structure and weren't random chains of amino acids. But proteins contain thousands rather than hundreds of atoms, so the methods that Hodgkin had used to solve vitamin B12 would not work. Fortunately, Max Perutz, an immigrant from Austria, decided to take on this daunting problem. Perutz had left Austria in the 1930s, escaping the Nazis by only a few years. Like Hodgkin, Perutz went to Cambridge to work with Bernal, who by that time was known as Sage because he seemed to know everything. Perutz arrived in Bernal's lab just after Hodgkin had left and began working on hemoglobin, a large protein in our blood that consists of 4 separate chains, each with an iron atom that carries oxygen from the lungs to our tissues. It was about 50 times larger than any molecule that had been solved by crystallography at the time, and people dismissed Perutz as crazy. Perutz himself had no idea how he was going to solve it. He would proudly show beautiful diffraction pictures from his crystals to his colleagues, but when they asked him what it meant, he would quickly change the subject. Luckily for Perutz, Bragg, who became the Cavendish Professor in Cambridge in 1938 and thus had great influence, was very enthusiastic about his goals and supported him for years, even though progress was slow or non-existent. Eventually, almost 20 years later, in 1953, Perutz made his breakthrough. When he added a heavy atom, like mercury, to his crystals, it changed the intensity of the spots. The heavy atoms bound to only a few places in the molecule, and by measuring the differences they caused in the intensities of the X-ray diffraction spots, it was possible to figure out where the heavy atoms were. They did this by carrying out the same Patterson calculations that Hodgkin had done, but this time on the difference in intensities between the crystal with and without the heavy atoms bound. The calculations would reveal where these heavy atoms were, which in turn would allow them to determine the phase for each spot and calculate a three-dimensional image of the molecule. Using exactly this method, over the next 6 years, Perutz and his former student, John Kendrew, solved the structures of hemoglobin and a smaller related protein that also carries oxygen, called myoglobin. So, around 1960, nearly 50 years after X-ray crystallography had revealed the structure of common salt with its chessboard arrangement of just 2 types of atoms, the technique could show what a protein with thousands of atoms looked like in 3 dimensions. The age of structural biology had begun. Perutz was Crick's PhD supervisor, and Kendrew, at least officially, was Watson's postdoctoral advisor. Perhaps not entirely by coincidence, Perutz and Kendrew shared the Nobel Prize in Chemistry in 1962 for the first protein structures, the same year that Watson and Crick shared the Nobel Prize in Physiology and Medicine with Maurice Wilkins for their work on DNA. That same year, Perutz moved his unit from the refurbished bicycle shed behind the Cavendish lab in the middle of town, where they had been tolerated by the real physicists for many years, to their new home in a new 4-story building in the southern outskirts of Cambridge, the MRC Laboratory of Molecular Biology, or LMB. With 4 Nobel Prizes in its first year, the LMB kicked off with a big bang. Chapter 4. The First Crystals of the Machine. As a result of the heroic efforts by Max Perutz and John Kendrew, people could see for the first time how the thousands of atoms in a protein came together to form precise structures. In their case, they could even see the iron atom that bound the oxygen molecule in myoglobin and hemoglobin. Crystals are basically ordered 3-dimensional stacks of identical molecules. At one extreme, if a molecule has just one atom, making a crystal of it would be like making a regular stack of spheres like billiard balls, fairly easy. But a large, irregular molecule with thousands of atoms will not stack up so easily because all the molecules would have to line up in exactly the same orientation. Any slight irregularity, and you would disrupt the pattern. It would be like piling up toy railway engines into a perfectly aligned and regular stack. The problem is even harder because large molecules like proteins are not entirely rigid. They tend to be floppy, with little loops and extensions that wave around. So it is amazing that proteins crystallize at all. And in general, the larger the protein, the harder it is to crystallize. Even today, nobody can predict exactly how a protein will crystallize, or even whether it will do so at all. Considering how uncertain the process is, it was not at all clear something like the ribosome, which had hundreds of thousands of atoms, not just thousands, would even form crystals. For molecules to form a well-ordered crystal, they need to be nearly identical, so they can all pack the same way in a 3-dimensional stack. People initially didn't know whether all the ribosomes from a particular source, like a bacterium or a tissue of an animal, had the same structure, or even the same set of proteins. If they didn't, it was highly unlikely that they would ever form crystals. The first hint that ribosomes might have a defined structure came only a decade or so after ribosomes were discovered, when Breck Byers at Harvard was looking at what would happen to cells in chick embryos when they were cooled. He wasn't initially looking for ribosomes at all, but was studying long filaments in cells called microtubules that are involved in lots of processes like cell division. While doing this in 1966, he noticed that ribosomes in these cooled chick embryo cells would come together in regular sheets. These sheets were only one ribosome thick, forming two-dimensional crystals rather than the usual three-dimensional ones. Max Perutz invited Byers to the LMB to work on his two-dimensional crystals. He visited twice in the 1960s and 1970s, but apparently nothing came of it. In the meantime, two young scientists at the LMB, Nigel Unwin and Richard Henderson, were figuring out a different way of determining the structure of biological molecules. Unwin was tall and thin with bangs that made him look like a beetle, while the shorter Henderson looked boyish enough to pass as a teenager, exacerbated by his tendency to wear shorts and sandals. Both were energetic and eager to make their mark on science. Unwin and Henderson were working out how to determine the structure of bacteria rhodopsin, a protein that sits in the membrane of a salt-loving bacterium and generates energy from light. At the time, there was no good way to get membrane proteins to form three-dimensional crystals. These proteins sit in the oily environment of the lipid membrane that envelops all cells and are therefore not soluble in water, so the traditional methods of crystallizing a protein would not work. Unwin and Henderson decided to work with two-dimensional crystals of the sort that Byers had seen and used the electron microscope to obtain their structure. Like X-rays, electrons have a wave-like character and have an even shorter wavelength. They had been used to obtain atomic structures of substances like minerals and metals, but biological molecules have very low contrast, meaning that in terms of their scattering properties, they do not stand out much compared to the surrounding water or lipid membranes. To see them with sufficient detail, you need to expose them to so many electrons that the molecules would be damaged and disintegrate before you could produce their structure. However, with two-dimensional crystals, Unwin and Henderson worked out methods of obtaining a structure by doing crystallography using an electron microscope with low doses of electrons. In 1972, while they were just starting to work out their methods, Unwin came across a paper reporting that ribosomes formed regular two-dimensional arrays similar to what Byers had observed, only this time from the oocytes, the cells from which eggs develop, of a kind of lizard. He wrote to the author, Carlos Today, inquiring about these crystals, but never received a reply, even after several attempts. With what could only be described as grim determination, Unwin took a train all the way from Cambridge to Naples, found Today's lab, and knocked on his office door. Eventually, Today came to the LMB for a while to work with Unwin. Apart from his curious reluctance to respond to Unwin's initial inquiries, he was eccentrically insouciant in other ways. He became well known in the LMB for blithely puffing away at his pipe in the lab and regularly setting off the fire alarms. Unwin pursued the problem for a few years, and although he did get some information, it became clear that these two-dimensional crystals of ribosomes from lizard oocytes were not good enough to obtain a detailed atomic structure. So Unwin eventually gave up on the problem and moved on to other things. Both he and Henderson went on to do pioneering work on the structure of membrane proteins. Unwin's lizards, which were kept in the basement of the building, escaped and multiplied, and could occasionally be seen wandering outside the building for years afterwards. Even though the two-dimensional crystals of ribosomes from chick embryos and lizard oocytes proved to be a dead end, they were important because the very fact that ribosomes could crystallize at all, even if only in two dimensions, suggested that at least they had a defined structure. Could the ribosome ever form three-dimensional crystals of the sort that had been used to solve the structures of proteins like hemoglobin? By the mid-1970s, many protein molecules much larger than hemoglobin had been crystallized, including large protein assemblies and entire viruses. So even though ribosomal subunits were 10 times larger than the largest molecule that had been crystallized to date, it was not so unreasonable to try to coax them into forming the kinds of crystals from which one day a structure could emerge. One person who thought so was Heinz Gunther Wittmann, who, with his wife Brigitte Wittmann-Liebold, had worked on the genetic code using tobacco mosaic virus, which uses a single RNA molecule rather than DNA to store its genes. In 1966, Wittmann was made a director at the new Max Planck Institute for Molecular Genetics in Berlin. This meant he had enormous resources and oversaw a department called by its German name of Abteilung. The papers from the department would have Abteilung Wittmann as part of the address. Nowadays, Max Planck directors rarely confer their own name on their department, instead preferring to call it after the area of their research. Once the Max Planck organization hired you as a director, it was virtually unheard of for them to fire you. This meant that Wittmann could try things that might take a very long time to work. In an almost stereotypically German way, Wittmann systematically organized his department to look at every aspect of ribosomes. Some of it was important at the time, but mind-numbingly tedious, like purifying ribosomal proteins from lots of different species and laboriously sequencing each of them. DNA sequencing, developed by Frederick Sanger in 1977, superseded much of this work because it was much faster to sequence the gene for a protein than to sequence the protein itself. But Wittmann was also smart enough to know that structure was the key to understanding how the ribosome worked. A few years after Wittmann started his department, a curious character appeared in the world of crystallography. He was a German named Hasko Paradis, and although a pediatrician by training, he had taken to working on crystallizing important molecules. It seemed as though there was nothing he had not crystallized. He had crystallized tRNA before anyone else, as well as many large protein complexes. The only problem was that his work did not stand up to scrutiny. When David Blow, a pioneer in crystallography of enzymes, saw an X-ray diffraction picture of one of the so-called tRNA crystals during a talk Paradis gave when he was working at King's College in London, he immediately recognized it to be chymotrypsin, a protein he had solved many years earlier. He confronted Paradis, and in fairly short order, Paradis left King's. On the strength of his having crystallized so many different important proteins, Paradis was offered a job in Wittmann's department, even though Wittmann was apparently told of the circumstances surrounding Paradis's departure from London. He stayed in Wittmann's institute until 1974, the year he published a paper on crystallizing ribosomes, and went on to a professorship at the Free University in Berlin. Some years later, in 1983, Wayne Hendrickson and several other leading crystallographers wrote a letter to Nature laying out the reasons for their conviction that key pieces of Paradis's research represented deliberate misrepresentation and should be discounted. Although he responded to the letter defending his work, Paradis left his post in Berlin soon afterwards for what he maintained were unrelated reasons. At this point, he disappeared from the world of biological structures. Often just being told something is possible breaks down a huge psychological barrier and spurs people to try things. So even though Paradis's claim of tRNA crystals was widely rejected, Brian Clark, who was working on the problem at the time, said he was spurred on by Paradis's claims and even found some of his suggestions useful in producing actual crystals of tRNA. Similarly, Wittmann, unaware or unconvinced of Paradis's failings and seeing his initial result as promising, continued to be interested in having someone try to crystallize the ribosome. One person who was game was Bob Fletterick, a crystallographer who was at the University of Alberta in Edmonton, Canada. He was about to get tenure there, but his girlfriend at the time was a native-born German, and he thought it would be good for them to spend a few years in Germany. So, in early 1978, he contacted Wittmann to discuss the possibility of working on crystallizing the ribosome. Wittmann agreed it would be worthwhile and sponsored Fletterick for a Humboldt Fellowship, which was granted some months later. Fletterick recalled the salary as being higher even than for the faculty position he had in Canada. His stint with Wittmann never materialized, however, because Fletterick's girlfriend suddenly took a diversion, so he no longer felt like going to Germany. By that time, he was getting lots of faculty offers in the US and ended up spending the rest of his career in San Francisco as a tenured faculty member at UCSF. Having struck out with both Paradis and Fletterick, Wittmann might have been feeling discouraged. But he was lucky the third time around when a scientist from Israel, Ada Yonath, inquired if she could work with him. She had exactly the right combination of ambition and tenacity needed for this project. At the time, Yonath was a faculty member at the Weizmann Institute in Israel. At some point after meeting Wittmann at a conference, she broached the idea of spending some time at his institute in Berlin. Fortunately, Wittmann still had the unused Humboldt Fellowship from his application with Fletterick, and he quickly arranged for it to be given to Yonath instead. Yonath's road to Berlin was not an easy one, and she had to overcome some daunting barriers along the way. She grew up in Jerusalem, in a poor Orthodox family whose circumstances were made more difficult by the premature death of her father at the age of 42. Yonath's parents encouraged her education, although her difficult circumstances meant that she had to work and help support the family from quite a young age. In an early glimpse of her determination, she managed to find the resources to go to university, went on to graduate from the Hebrew University of Jerusalem, and obtain a PhD from the Weizmann Institute. After doing a postdoctoral stint in the US, she returned to the Weizmann Institute to become a faculty member there. Yonath began her ribosome-related work by trying to crystallize one of the protein factors that helps the ribosome start at the right point on the mRNA. But after a year, she had had little success with it. She suffered a setback after a bicycling accident and had to spend several months recuperating. During this time, according to an interview with Elizabeth Panizzi in Science in 1999, she realized that Wittmann's lab was making lots of purified ribosomes from different species and asked Wittmann if she could try to crystallize them. Yonath's experience in crystallography was limited to a couple of small proteins, and she had no previous publications related to ribosomes. But after his 2 false starts with Paradis and Fletterick, Wittmann was probably only too delighted that someone was willing to take on the extremely challenging project. In the same Panizzi article, Yonath recalled, "He said this was the dream of his life and gave me everything I needed." Advances in biology are often made by choosing the right organism to work on. For example, the study of nerve transmission was made possible by using giant axons from squid because they were large enough to see and stick electrodes into. Early geneticists used fruit flies because they breed quickly, and you can follow lots of visual markers like eye color to deduce how various traits are inherited. For bacteria, the standard organism for biochemical and genetic studies of all sorts is Escherichia coli, or E. coli for short, because it is easy to grow and doubles every 20 minutes, and one could do genetics with it. Its name refers to both the person who discovered it, Theodor Escherich, and who it's being found in the human colon. It is more familiar to the general public because of the occasional outbreaks of severe dysentery some virulent strains of it can cause. Not surprisingly, it became the main source for purifying and studying ribosomes, and Wittmann's lab had an abundant supply of the microorganism. The initial efforts to obtain crystals from E. coli ribosomes resulted only in small microcrystals, so small that they were no more useful than the 2-dimensional crystals others had already been working on with electron microscopy. They needed a new organism, and luckily, their colleague Volker Erdmann had just the right one to get them started. At age 15, Erdmann immigrated from Germany to the US, where he went to high school and college in New Hampshire. He was curious to return to his roots, so he went back to Germany for his PhD. After his PhD, he went to work in Masayusa Nomura's lab in Wisconsin. He had heard of Nomura's work on taking apart and putting back together the entire small, or 30S subunit, of the ribosome, and wanted to see if he could do the same with the large, or 50S subunit. The names 30S and 50S for the subunits of bacterial ribosomes indicate how fast they sediment in a test tube when spun at high speeds in a centrifuge. The capital letter S stands for Svedberg units, after the Swedish scientist Theodor Svedberg, who characterized how molecules sediment in an ultracentrifuge. Oddly, the whole bacterial ribosome is 70S, not 80S, because how fast a particle sediments depends on its overall shape as well as its mass. Erdmann initially tried to reassemble 50S subunits from E. coli, which is naturally what Nomura had used for the 30S subunit, but he failed completely. So he switched to a bacterium called Bacillus stereothermophilus. The name stereothermophilus means heat-loving, and the bacteria grow naturally in hot springs at around 60 degrees Celsius or 140 degrees Fahrenheit. When he took 50 S subunits from these thermophilic bacteria apart, he was able to repeat the trick Nomura had done with the small subunit. After his fellowship, in the last of his oscillations between Germany and the US, he moved to Wittmann's department in Berlin, where he set up his own lab and took his samples of 50 S subunits with him. One day in the late 1970s, Jonath and Wittmann told Erdmann about their plan to crystallize ribosomes and asked him if he would like to help. Erdmann said he could help them if they worked with B. stereothermophilus ribosomes, since he was familiar with them. Because molecules from thermophilic bacteria are resistant to heat, they are thought to be more stable and likely to crystallize. They decided to meet the following Sunday morning. Erdmann asked his wife, Hannelore, who worked with him, to come along because she knew where the old samples of the large subunit were in the freezer. Jonath set up crystallization trials on them with Erdmann and his wife. Just 3 days later, on Wednesday, Erdmann recalled, Wittmann told him they had crystals. Barent Tescher, an electron microscopist, confirmed that they were indeed crystals of the 50S subunit. The team tried to improve on the original small crystals. When Erdmann's original material ran out, they had to make large subunits from freshly made ribosomes. There was a period when they couldn't reproduce the original crystals. Erdmann told me jokingly that he worried that since the original crystals were from subunits that had been stored in a freezer at minus 80 degrees Celsius for 4 years, they would have to wait a few years each time after they purified ribosomes to obtain crystals. In the end, however, they managed to get crystals routinely. Getting real three-dimensional crystals of a molecule with several 100,000 atoms was a major achievement. But instead of reporting it with great fanfare in one of the major journals, the results were published in a then new and now defunct journal called Biochemistry International. Wittmann was a founding editor of the journal, whose name belied the fact that hardly anyone read it. I asked Erdmann why Wittmann would choose a relatively obscure forum to publish such an important result instead of a high-profile journal like Nature or Science. He speculated that Wittmann was fairly conservative and also a little hesitant and cautious because of the Parodies episode, so he didn't want to make a big deal of it, but wanted to at least put it out on record. Buoyed by their initial success, Wittmann did his best to ensure long-term support for Jonath to continue working on the problem. He tried to get her a director's position like his own, but the august Max Planck Society, apparently unimpressed by Jonath's credentials at the time, declined. However, he did eventually persuade the society to support a special lab for her in Hamburg, right next to the German synchrotron, whose powerful X-ray beams would be needed to study these crystals. Just as importantly, he provided extensive support from his own department for making and characterizing the ribosomes. Over the years, he and Jonath became close friends. Their success motivated others to enter the fray. The small town of Pushchino was built by the Soviet government as a center of science. It was home to several well-funded institutes, one of which was headed by a brilliant ribosome biochemist named Alex Spirin. Like Wittmann, Spirin too was directing a large group studying virtually every aspect of ribosomes. He was not as systematic as Wittmann, but was a highly imaginative scientist who would publish bold new ideas because he was unafraid to be wrong occasionally. Spirin was also a highly independent man who didn't like to bow to authority. One example of this was when he was asked to sign a petition to expel Andrei Sakharov, the dissident nuclear physicist and father of the Soviet hydrogen bomb, from the Soviet Academy of Sciences. Openly refusing to sign the petition might have been politically awkward for a prominent member of the Academy who was also the director of a major institute, so Spirin decided to become unavailable by going on a long hunting trip in some remote forests outside Pushchino. Spirin's institute was also interested in the structure of ribosomes, and one member, Maria Garber, ran a small group that was trying to crystallize individual proteins of the ribosome or the protein factors that help the ribosome do various tasks. Like everyone else, she too tried proteins from E. coli ribosomes. Garber changed the course of ribosome research in 1978 when she read a report from Japan that described using a new bacterium that grew at even higher temperatures than B. stereothermophilus as a source to crystallize two important proteins that acted on the ribosome. It was isolated from the hot springs of the Izu Peninsula in Japan in 1971 by Taira Oshima and grew best at the searingly hot temperature of 75 degrees Celsius or 167 degrees Fahrenheit, which would only take a few seconds to burn your hand if you were so foolish as to plunge it into a hot spring at that temperature. They gave the new bacterium the unmistakable, if tautological, name of Thermus thermophilus. Garber decided that this new bacterium was the answer. She went to Japan for a few months and brought back some stocks of the bacteria in December 1979. But unfortunately, they had died en route. She asked Toshima to send her some fresh cells by mail, which arrived safely. By the end of 1980, Garber and her colleagues had successfully used these bacteria to obtain beautiful crystals of a large protein, or factor, called elongation factor G, which helps the ribosome move along the mRNA. This initial success with T. thermophilus proteins encouraged Garber and her colleagues to try other things with the organism. Given the resources in the Soviet Union at the time, it was quite expensive to grow large amounts of T. thermophilus, and Garber didn't want to waste any of it. They invited other scientists from the USSR to take whatever proteins they wanted from the bacteria, like a slaughterhouse, using every part of an animal that is killed. One of Garber's colleagues was Igor Serdyuk, a Communist Party member involved in communication with foreign countries, who had no problem traveling frequently to the West, even at the height of the Cold War. He had previously used low-resolution techniques to characterize the overall shape of ribosomes, so he was naturally interested in seeing if he could crystallize them with material from Garber's laboratory. He and his student, Liza Karpov, purified ribosomes from T. thermophilus and obtained very small crystals, like the very first crystals in Berlin. With this initial success, Garber asked Spirin if he would support a group to try to crystallize ribosomes from this new organism. He agreed, and she was joined by several people, of whom the most notable was Marat Yusupov, a student of Spirin's. Since they didn't have a lot of crystallization expertise, they asked two people from the Institute of Crystallography in Moscow to help them, Vladimir Baranin and Sergei Trakhanov. By 1986, they had crystals of both the small subunit and, using a trick Trakhanov had used to purify the ribosomes, the entire ribosome. Along with the 50S crystals from Jonath's and Wittmann's efforts, this meant that both subunits and the entire ribosome had now been crystallized. Yusupov presented their results as a poster at a meeting in July 1987 in Buchenberg, near Strasbourg, France, and a month later, the work was published in the European journal FEBS Letters. Within a few months, Jonath and Wittmann reported that they too had crystals of the small subunit and whole ribosomes from the same T. thermophilus species that the Russians had used. They published their results in the same obscure Biochemistry International journal where they had reported their original crystals several years earlier. The following year, Jonath reported improved crystals of the small or 30S subunit that looked at least as good as the Russian ones. This might have led to a head-to-head competition between the Russian and German groups, but it was not to be. In comparison with the Germans, the Russians were poorly funded and equipped, especially for crystallography of large molecules. In an effort to take the work to the next stage, Marat Yusupov and his wife, Gulnara, went to Strasbourg to continue the ribosome crystallography work in collaboration with Jean-Pierre Ebel and Dino Morat. For reasons unknown to Yusupov, at some point, Ebel decided to discontinue the collaboration. For his part, Spirin believed that Ebel was discouraged by Jonath and Wittmann from competing with them. Whatever the reason, the Russian effort on crystallization of whole ribosomes fizzled out, and Maria Garber went back to her original interest in individual ribosomal proteins and factors. With their efforts frustrated, some of the key people in the Russian effort dispersed all over the world. Some years later, in the mid-1990s, Yusupov wrote to Harry Noller, a leading ribosome biochemist at UC Santa Cruz in California, asking to work on the structure of the ribosome in his lab, but that is a story best told later. Sergei Trakhanov seems to have led a peripatetic life, doing various jobs in Japan and the US for almost 2 decades. He too worked for some time in Noller's lab, after Yusupov had left it, before returning to Europe. With the Russian effort essentially closed down, Jonath was leading the only group working on ribosome crystallography. By the end of the 1980s, none of the crystals was good enough to yield an atomic structure of either subunit of the ribosome, let alone of the entire object. But they could, in principle, have been good enough to reveal something about how the proteins and RNA were organized. Instead, progress on the structure of the ribosome was slowly emerging from the fuzzy images of the ribosome seen by electron microscopy. Some of the work involved antibodies, which are proteins produced by our immune system that can bind to very specific targets. In one clever experiment, Jim Lake from UCLA, one of the scientists studying ribosomes using electron microscopy, made antibodies that recognized the beginning of a newly made protein. He showed in 1982 that these antibodies were attached to the backside of the large subunit. This was on the opposite side from where the new amino acid on the tRNA was attached to the growing protein chain. This suggested that there must be a tunnel in the large subunit, a birth canal, through which a newly made protein chain would have to travel before it emerged on the other side. A few years later, in 1986, Nigel Unwin confirmed the presence of this tunnel by analyzing his two-dimensional crystals of lizard ribosomes in the electron microscope. The following year, Jonath and Wittmann also reported that there was a tunnel by analyzing two-dimensional sections of their crystals of ribosomes in the electron microscope. Both of these reports were of fuzzy or low-resolution structures. Also, neither the ribosome nor the tunnel in these structures bear a strong resemblance to the ribosome we know today. It is not at all obvious that either group would have been so confident in identifying features in their maps as the tunnel through which the newly made protein chain emerges if Lake had not already shown it existed. Apart from these limited results, progress was slow. Even a decade after the first three-dimensional crystals of the ribosome had been produced, it was not obvious that they could ever be used to produce any meaningful images by X-ray crystallography. Certainly, an atomic structure of the ribosome seemed like a pipe dream. Nevertheless, even when it was not at all clear that it would ever be technically feasible, Ada Jonath kept the dream alive by continuing to explore different species and conditions to improve her crystals. Chapter 5. Going to the Mecca of Crystallography. Meanwhile, in the mid-1980s, I was slowly becoming frustrated at Brookhaven, trying to milk what I could using the techniques I knew. They would never produce the kind of detailed images that could lead to an understanding of how the ribosome worked or even how chromatin was organized. Luckily, a few years after I got to Brookhaven, I was joined by Steve White, who had come from Wittmann's department in Berlin. Steve had grown up in a working-class neighborhood in East London, and the odds were stacked against his emerging out of it. But he was smart and lucky enough to study at a grammar school, a selective government-funded school in England that admits pupils on the basis of an entrance exam taken at the age of 11. From there, he went on to get an undergraduate degree from Bristol and a PhD from Oxford. He was a sociable and friendly man who shared my bawdy sense of humour and, like many Englishmen, enjoyed a beer with friends and liked sport of all kinds. When he arrived from Berlin, he was involved with a South Indian woman who had moved to Philadelphia, but the relationship that had endured a transatlantic separation did not survive the much shorter distance to Long Island. With its abysmally low ratio of women, Brookhaven was not exactly promising for the dating scene if you were a single guy. But somehow, Steve, with his English accent, outgoing manner and charm, managed to enter into a series of relationships during his time there. In Berlin, instead of working on entire ribosomal subunits like Ada Jonath, Steve and his fellow Englishman Keith Wilson were working with Christoph Appelt and others on the much more feasible goal of solving the structures of individual ribosomal proteins. After a long hiatus, they'd solved the second one, leaving only 48 more to go. But even if they had solved all of them, it would be like knowing what lots of the peripheral parts of a car looked like, such as a fuel line or a spark plug, without any idea of how they fit together in a working vehicle. Of course, the 2/3 of the ribosome that was RNA would still remain completely unknown, but it was better than nothing. I suppose what we hoped was that the structures would provide some clues about how they fit into the ribosome and what they did. Steve was keen to carry the project forward after he arrived there. Steve was a well-trained crystallographer who had already been involved in studying several ribosomal proteins. I, on the other hand, knew a lot about how to take a ribosome apart and purify its proteins, but knew no crystallography at all. Given our shared interest in the ribosome, Steve suggested we work together. I was delighted, and thus began a collaboration that lasted almost 15 years and changed my life. Initially, I went to Yale to use their giant fermenter to grow large amounts of the same Bacillus stereothermophilus bacteria from which the Berlin group had obtained their first 50S subunit crystals, and from which Steve and his colleagues had crystallized several ribosomal proteins. But after seeing how tedious the whole procedure was, and how little pure protein we got from it, I felt there had to be a better way. As luck would have it, I was in exactly the right place at the right time. My colleagues, Bill Studier and John Dunn, were busy figuring out how to trick the standard E. coli bacteria into making large quantities of almost any protein they wanted. They did this by using a special signal used by a virus called T7 that attacks E. coli and enables it to hijack the bacterial apparatus to make its own proteins. They figured that they could get E. coli to make large quantities of any desired protein by introducing the special signal from T7 to the beginning of the gene for that protein. So I told Steve I was going on a little sabbatical in our department to learn the tools of molecular biology, and he should just wait for a while. My lab at the time consisted of 2 technicians, Sue Ellen Gertschmann and Vito Graziano, who were joined a few years later by a third, Helen Kaiser. Under Bill and John's tutelage, Sue Ellen and I quickly cloned the genes for all the proteins that Steve White and his Berlin colleagues had crystallized. Soon after that, we made lots of the proteins and were delighted when these genetically engineered or recombinant proteins made in E. coli crystallized exactly like the ones purified by direct extraction from ribosomes from the original thermophilic bacterium. At this point, I was also working on a key protein in chromatin that helps to condense it into a filament and pack it in the nuclei of cells. This protein, called a linker histone, has a central core called GH5. I wanted to crystallize it, but had no experience in how to obtain crystals. Steve said, "It's very straightforward. Let me show you how." Any school child knows that if you let a solution of salt or sugar dry out, you get crystals. This is because as it dries out, the salt or sugar becomes so concentrated that it is no longer soluble, and the molecules pack together to form crystals that come out of solution. If you let proteins dry out, though, they just form an amorphous goop, not crystals, because they are large and floppy, and there are just too many ways for them to pack against each other. This is what happens when you add lemon juice to milk, and the proteins quickly curdle. To coax large protein molecules to come out of solution in the form of crystals, you have to increase their concentration very slowly, giving the molecules the opportunity and time to line up in an orderly way to crystallize. This was typically done by mixing a drop of a protein solution with a small amount of precipitant, like alcohol or salt, which makes the protein insoluble. This drop is then placed on a thin glass slide called a cover slip and inverted over a little well containing a solution that has a lot more salt or alcohol or whatever it is that makes the proteins insoluble. Water molecules leave the drop as vapor and enter the solution in the well until the salt concentration is the same in both the well and the drop. As the drop slowly shrinks because water is leaving it, the precipitant and proteins become concentrated enough to make the proteins insoluble. If it is done slowly enough, and if the conditions are just right, the proteins will come out of solution by packing regularly against each other to form crystals. In those days, we had to do all of the manipulations by hand. But today, robots routinely set up thousands of trials by varying the composition of the protein solution to try to find a condition that produces crystals. Steve helped me set up a few different ways of trying to get crystals, and we soon had crystals of GH5. In the meantime, we had also reproduced crystals of a ribosomal protein called S5. The prefix and number meant it was roughly the 5th largest protein from the S small subunit. Having obtained crystals of these proteins, I didn't want to just sit back and watch Steve solve them. I wanted to learn how to solve them myself. But I didn't know any crystallography, nor did I know how hard it would be to learn it, let alone become expert in it. Steve was reassuring. With your background in physics, you'll find this stuff dead easy by comparison. That gave me the courage to embark on yet another venture. But how to go about it? For a start, I took a crash course in crystallography at Cold Spring Harbor Laboratory, a well-known lab just 30 miles west of Brookhaven that was headed by none other than Jim Watson. In addition to conducting research and organizing meetings, the lab ran short, specialized courses for scientists to pick up new techniques, often taught by world-renowned experts. In 1988, they had just started a new 2-week course in crystallography, and I figured it would be a great way to quickly acquire the basics of the method. I was taught by some of the greatest scientists in the field, and on a free day, I took one of them, Hans Diessenhofer, on a long walk to see Teddy Roosevelt's home a few miles to the north of the lab. Hans was wearing totally unsuitable dress shoes and developed painful blisters on the way back. He probably forgot about his pain 2 days later when he shared the Nobel Prize for determining the structure of the protein complex that captures energy from sunlight and converts it into chemical energy, one of the most fundamental reactions of life. A year later, my department was considering whether to give me tenure. If they didn't, then I would be out of a job, and learning crystallography would be the least of my worries. I had produced a few good papers, mostly using neutron scattering, but by that time, I had already concluded that the technique produced almost no information of real relevance to how molecules actually worked. My work had reached a dead end, and crystallography was clearly where the action was. Hardly a week went by without a new article on how the atomic structure of some important molecule had forever changed the understanding of that field. Moreover, the tentative initial forays I'd made with Steve's help and the course I'd taken had whetted my appetite. A tenure committee grilled me on my long-term plans. I took a deep breath and told them that, if they gave me tenure, I'd stop doing what I'd been doing, which was using neutron scattering, and immediately go away on sabbatical for a year to learn crystallography. To my immense relief, they agreed it was a good idea. A few days later, the senior staff of the department met, and that evening, John Dunn came to my home and handed me a long stick covered with aluminium foil, saying, "Welcome to the tenure staff." When I began thinking about where I should go on sabbatical, there was really only one place I considered, the LMB in Cambridge. It was the birthplace of protein crystallography, and of course Cambridge was the birthplace of all crystallography. I also knew lots of Americans had gone there on sabbatical and enjoyed it. On a personal level, Vera and I were Anglophiles who loved English literature and culture. We religiously watched Masterpiece Theatre, and I loved the eccentric off-the-wall humor of Monty Python. The director of the LMB at the time was Aaron Klug, a towering figure in structural biology. Aaron had led one of the 2 groups that solved the structure of tRNA, the other being a collaboration led by Alex Rich at MIT and Sung-Hoo Kim at Duke. As is often the case with high-stakes competition, that race to obtain the first tRNA structure ended up being quite acrimonious. Aaron was a protege of Rosalind Franklin's and at first sight could be mistaken for Woody Allen. He was also a leading figure in chromatin structure, and I thought it would be great to work in his lab. So I summoned up my courage and wrote to Aaron, saying that I had crystallized the linker histone involved in compacting chromatin and would like to come to the LMB on sabbatical to learn crystallography so I could solve its structure. I was pessimistic that he'd be interested in a nobody like me, but was delighted when he wrote back a few weeks later encouraging me, saying he would be happy to sponsor me for a Guggenheim Fellowship. With a fellowship and a half salary from Brookhaven, I was all set to go off to England for a year. I didn't want to embarrass myself with Aaron after he had taken a chance on me, so I thought I should go prepared with some real data that I could learn to analyze on my sabbatical. Despite the crash course in Cold Spring Harbor, I really didn't know the details of collecting and processing data, but here another colleague came to the rescue. Bob Sweet had grown up in small-town Illinois before going to Caltech for his undergraduate work. After his PhD from Wisconsin, he went to the LMB to do a postdoc, like so many Americans, before spending time as a faculty member at UCLA. Like many Midwesterners, Bob too was an Anglophile, and his stint in Cambridge had only made it worse. He would routinely use anglicized vocabulary, grammar, and spelling. Some years earlier, he and I had arrived at Brookhaven the same week and were living in temporary housing on site. When I first saw him, I was instantly struck by his large, luxuriant, and meticulously tended Poirot-like mustache. As he became bolder over the years, it became an even more prominent feature. He could be sarcastic, with a pedantic, avuncular manner that put some people off, but I found him a thoughtful, warm, and generous person. We went on to become good friends. Bob ran a crystallography instrument, or beamline, at the X-ray synchrotron at Brookhaven. He took me under his wing and taught me a huge amount about how to collect and process data from crystals. Bob also got me interested in a crystallographic technique that turned out to be crucial later for solving the ribosome. Ever since Max Perutz and John Kendrew had solved the first protein structures 30 years earlier, the main way to solve structures was to collect data on the protein crystals and then repeat the process by soaking different compounds of heavy atoms like gold or mercury into the crystals. The differences between the data with and without the heavy atoms would reveal their positions and allow you to determine the phases of the X-ray reflections. With these phases and the measured intensities, you could calculate the structure. Getting crystals of a protein was uncertain enough, but getting these heavy atom derivatives introduced yet another uncertainty. Often, soaking heavy atoms would destroy the quality of the crystals, or they wouldn't bind to the proteins at all. The heavy atom method became known as soak and pray. But just as I started doing crystallography in the late 1980s, a new method called multi-wavelength anomalous diffraction, or MAD, began to produce some promising results. The Dutch crystallographer Johannes Bevot had established the principles of MAD in 1949. It was based on the fact that some atoms can absorb and re-emit X-rays instead of scattering them right away. As a result of this anomalous scattering, there are small differences in the intensities of pairs of diffraction spots that should be completely identical because of the symmetry of the crystal. The pairs of symmetric spots are called Friedel pairs, and the difference in their intensities, the anomalous differences, contain phase information, just like the differences as a result of adding a heavy atom to a crystal. Normally, atoms in a biological molecule like carbon, nitrogen, or oxygen do not have much anomalous scattering, so any differences are typically too small to be useful. But in 1980, Wayne Hendrickson, then at the Naval Research Lab, had solved the structure of a small protein using just the anomalous scattering from the unusually large number of sulfur atoms in the structure, which came from the amino acid cysteine. Around this time, synchrotrons started being used for X-ray crystallography. They are large particle accelerators that accelerate electrons to close to the speed of light. As the electrons orbit around, they emit extremely intense beams of X-rays that could be used for diffraction studies. Keith Hodgson and his colleagues at Stanford realized that, with synchrotrons, you could also choose the wavelength of the X-rays incredibly precisely. This meant you could collect data at two different wavelengths, where the scattering of some special atoms would change significantly. The difference between the two datasets would give you the position of the special atoms, and you could then calculate phases just as you might with heavy atoms. Moreover, you could do this in a way where at one of the wavelengths, the anomalous scattering of the special atom was particularly large. Wayne Hendrickson developed an elaborate formalism to do this that was different from the one Hodgson had outlined and went on to solve a series of structures with it. Wayne also came up with the brilliant idea of growing bacteria in which the sulfur atom in the amino acid methionine was replaced with selenium, so that all the proteins would have selenomethionine wherever there would normally be a methionine. Not only was the anomalous scattering of selenium much larger than that of sulfur at typical X-ray wavelengths, but it had a peak at a wavelength that was very convenient for synchrotron instruments, around 1 angstrom, or 0.1 nanometers. This made the method incredibly powerful. In principle, any protein that had a sufficient number of methionines ought to be solvable this way, and today it is one of the most common ways of solving a brand new protein structure. Bob was interested in getting the method to work on his instrument at the Brookhaven synchrotron. So Vito Graziano produced crystals of GH5 with selenomethionine, and with Bob's help, we carefully collected data around the wavelengths where the selenium atom changed its properties. Steve and I also collected data on S5, but by the traditional heavy atom method, where we had soaked a gold compound into the crystals. I now had complete data on 2 proteins, but no idea what to do with them. It was time to head for England. My family and I took off for England in late August 1991. When we landed, I rented a large people carrier and packed the 4 of us and all our luggage, including 3 bicycles, into it. Despite having to drive on the left side of the road after an overnight flight from New York, I managed to find my way to Cambridge without an accident. When we got to the Addenbrooke's Hospital site, I was lost in its labyrinthine maze of one-way streets and asked passers-by where the MRC Laboratory of Molecular Biology was. To my great surprise, the first few people I asked had no idea where the world-famous lab was. This immediately reminded me of the story in Crick's autobiography, What Mad Pursuit, in which his taxi driver had never heard of the Cavendish lab, even though scientists the world over had known it for over 100 years. It made me realize that fame among scientists has a very narrow reach. Aaron had assigned his longtime colleague John Finch to be my official host. Like Aaron, John too had been in Rosalind Franklin's lab and had moved with Aaron to Cambridge when the new MLB building had opened. When I arrived, John told me that, unfortunately, they did not have a place for me to work just yet. I rather naively told him that all I needed was a small desk in a corner of his lab. John politely smiled at this, and I found out a day later that he, a world-famous scientist, only had a desk and a small part of a lab bench. This was typical of the LMB at the time. Many senior scientists didn't have their own lab, and often just had a desk in a shared lab or office. The lab was incredibly crowded, with equipment in the hallways and almost no free space. Crowding people together may well have been responsible for the LMB's success, because it made people talk to each other and share ideas and techniques. On my first full day there, I arrived around 9 a.m., and about an hour and a half later, John came by and asked me if I would like to go up to the canteen for coffee. I thought I had hardly done anything yet and declined, saying I didn't drink coffee. Again, John gave me one of his enigmatic smiles, and a colleague who watched this exchange said, "He hasn't learned our ways yet." As the days passed, I realized that these regular breaks from work to have meals or coffee or tea allowed scientists to talk informally in the famous canteen on the top floor, a feature that has since been replicated in lots of scientific buildings. Humans can only really concentrate hard for a couple of hours at a time, and these short breaks re-energized people. The canteen was especially wonderful for a sabbatical visitor, because I quickly got to know lots of scientists and made several lifelong friends. My year at the LMB made me realize what a special place it was, and changed my entire outlook on science. Not surprisingly, many scientists all over the world view it as a model for how science should be carried out, even if they don't always persuade their own institutions to adopt its character. A notable exception is the Genelia Research Campus of the Howard Hughes Medical Institute, which explicitly modeled itself on the LMB and Bell Labs. I found that unlike the vast majority of scientists, almost nobody at the LMB was working on routine problems just because they would lead to publishable results. Rather, they were trying to ask the most interesting questions in their field and then developing ways to answer them. A simple but telling question they would ask each other was, "Why are you doing this?" Another lesson was that even very famous scientists like Max Perutz or Aaron Klug would unabashedly ask questions at lectures that were often trivial to people in the field. It made me realize that I shouldn't be ashamed of my ignorance, and that no question is too stupid to ask if you want to know the answer. A third lesson was that a lot of the LMB's success had to do with limiting the size of teams to just a few people. This forced the group leaders to focus on the most interesting questions and also participate, or at least stay in close touch with the actual work. Today, there is often a tendency for famous professors to build up huge groups of 20 to 30 people simply because they can. This may be great for the professor, But it is not often a great environment for the people being trained, because many of them are relegated to problems of lesser interest and have less frequent mentorship. Not surprisingly, many studies also show that these very large groups are actually less productive for the cost than smaller ones. In my first meeting with Aaron, he told me he thought the structure of GH5 wouldn't be that interesting by itself, and suggested I do some experiments to get it to bind to a piece of DNA. I really wanted to learn crystallography, but, being in awe of his reputation and too afraid to contradict him, I started working on his suggestion, sharing a lab bench with his postdoc, Wes Sundquist. After about a month, I realized that this was not something that could be done on the timescale of my sabbatical, if ever. With some trepidation, I told Aaron why I thought this was not a useful approach, and that I wanted to focus on solving the two structures using the data I had brought with me. Somewhat to my surprise, he readily agreed, and I think it increased his respect for me. Wes was amused as he watched an experiment I had abandoned slowly dry out and gather dust during the rest of our stay there. Even if he'd had the time, Aaron could not have taught me the nuts and bolts of how to solve a crystal structure, because the computing software had changed a lot since his day. Luckily, the LMB was, and remains, one of the most collegial places I have ever seen. Both junior scientists, like Paul McLaughlin, and well-known crystallographers like Andrew Leslie and Phil Evans took me under their wing and taught me the practical ins and outs of the method. Pretty soon, I was looking at a detailed map of S5 and building an atomic model of the protein. The excitement of building a structure cannot be exaggerated. Until that point, the molecule is a black box. You know it exists, and you know what it does, but not a lot more. Now, suddenly, it is as though the curtains have parted, and you see the molecule in its full glory, all of its atoms in their place, revealing the twists and turns of the chain as it folds up into its unique architecture to show how it might work. It must be how explorers felt when they came across a completely new landscape. Solving my other structure, GH5, had an interesting twist. Nearly every structure solved using MAD up to that point had used an elaborate formalism developed by Wayne Hendrickson. It used complicated bookkeeping to keep similar measurements made at different wavelengths separate and was cumbersome to use. More importantly, it didn't have the kind of sophisticated treatment of errors that standard crystallographic programs had. But we all thought that the special treatment was needed because the signal was so small. A typical gold or mercury heavy atom had about 80 electrons, but the typical variation in the properties of selenium from one wavelength to another in an MAD experiment was only a few electrons worth. It was a miracle the method worked at all, because the difference of a few electrons in scattering power was like adding a water molecule to the structure, hardly expected to make a difference. Using Wayne's elaborate programs, I had obtained what I thought was a decent map of GH5 and started building a structure of it. One day, Phil Evans returned from a visit to York, where his friend Eleanor Dodson thought a new program for solving standard heavy atom structures might also be useful for solving MAD structures. I was skeptical, but gave it a shot. To my surprise, within a few hours, I had a much better map, which we used to solve and publish the structure of GH5. After our paper, virtually nobody used Wayne's programs anymore, and instead began solving MAD structures with the same software they used to solve their other structures. If the signal from a MAD experiment is so small, why did the standard software work at all? I started thinking about it and realized, perhaps belatedly, that MAD worked even though the basic signal was small because the errors in the experiment were even smaller. What matters is not just the strength of the signal, but how much larger it is than the error or noise in the data, or what scientists call the signal-to-noise ratio. In the standard heavy atom method, when you collect data on crystals with and without a heavy atom, the two crystals are never identical. They have different shapes and vary slightly. To get a proper difference between them, you have to make sure all these factors are compensated for, so that the two sets of data are on the same scale, which is quite hard. Another problem is that adding a heavy atom changes the rest of the structure, a problem called non-isomorphism. This means you can never accurately compare the two datasets. In MAD, these problems don't exist. Data from the two wavelengths are collected on the same crystal, and the anomalous differences between symmetry-related spots are measured not only on the same crystal, but at the same wavelength, often at the same time. So, the signal-to-noise ratio in an MAD experiment is very good, and it typically produces much better maps that are easier to interpret in atomic terms. The full implication of how powerful anomalous scattering was didn't sink in right away. I had no idea how important a role it would play in my future. At the time, I was just pleased not to have made a fool of myself in my sabbatical among towering figures like Aaron. On the contrary, I had accomplished exactly what I wanted during my year in Cambridge, and the two structures I solved were published in Nature soon afterwards. Moreover, I had made a lot of friends and connections. What I didn't quite realize then was that my sabbatical had changed forever both my attitude towards science and how I would approach the ribosome. When I returned, I found I was no longer satisfied with making solid but incremental advances, and instead wanted to attack the big questions in the field.
So we are going to be shutting down now, and we'll recontinue this later on. Perhaps we're 2 hours and 20 minutes in, a little under 6 hours left of Venki Ramakrishnan's Gene Machine. I am blown away. I mean, there's a couple of personal stories which are interesting, but excuse me. All the discussion of X-ray crystallography. Finally, a sensible explanation as to why. Excuse me, there's been so much connection with Cold Spring Harbor. The use of heavy water. I'm blown away. The discussion of the role of big of computing in this. I just don't even know what to say. There's been a lot of great stuff. I'm going to have to go back, listen to this too, maybe 3 times. A little bit like the lab 257 from Plum Island. I just think that there's a lot of details in here, lots to learn. It doesn't feel as though so far there's any attempt to hide anything, and this is great. I mean, this is what we want to do. This is what I want to be, the channel to be representing. I do believe that there are a lot of great scientists out there, and boy, I'm just thoroughly impressed so far from what I've heard. If you want to direct message me, if you think otherwise, just let me know. But I'm really, really enjoying this book, and I look forward to the next 6 hours. I do know that he does talk about Wes Sundquist, I think, from Utah. He was the boss and a mentor of Jennifer Garris, who we've mentioned before, who was in the laboratory of Sina Bavari. I think that there's I'm just totally blown away so far. Great book. I hope you are enjoying it. And we will touch base again later on. I turned the camera around, but I didn't set it up properly ahead of time. And now I have to run some family errands. Every afternoon, new life here. So that's all, everyone. Thank you very much. Take care. Check back in later. We'll post this on the other channels, and God bless. I don't know why the exit didn't play there. Huh. Weird. Okay. Well, we're just going to close out then. Very strange.